All right, let's take a look at how you do dihybrids. So di means two, and a hybrid cross that's a dihybrid that you're using a Punnett square has to be not just a 4 square Punnett square, but 4 times 4 is 16 Punnett square. So you're going to be setting up a huge Punnett square because you're crossing two different characteristics, and of course each of the characteristics themselves has opposing potential traits. Okay, so it's going to be a big box. Part of the people's problems that people who get confused, part of the problem is figuring out how to set up the problem in the first place. The first thing you should do is have a key. So let's say that I have height as one of my characteristics, and let's say the other one is, I don't know, let's say color. So for height, we'll call tall dominant, and we'll call short recessive. So I'll make tall capital T, I'll make short lowercase t. It's good to pick a letter of the alphabet that has an uppercase and a lowercase letter that looks pretty different. One other trick though is if you pick something like an S, you could do a cursive S for the lowercase, and that's a way to tell them apart. But anyways, let's say color, I'll go with black and brown, and I'm going to say that black is dominant, and I'm going to pretend that brown is recessive. So the way you have to do these problems is basically FOIL. So in other words, first you have to figure out what the genotypes are, and then you FOIL them. So if I have a tall parent who we'll say is homozygous for tall, then I know that tall parent is going to be a big T, big T. But if that same parent is heterozygous for color, so like for instance they are black, but let's say they have a brown parent, then it would be big B, little b. What you need to do is you need to, when I say FOIL, you'll put them together, big T, big B, little b, and then you'll figure out which ones go together. So you'll do a big T and a big B. You'll do a big T and a little b. You'll do a big T and a big T and a big B. And you'll do a big T, little b. But that's just one parent. So now you got to figure out what the other parent is. So let's pretend that the other parent is a short parent and brown parent. I'm just going to make it simple because that'll be easy, because we'll just have short as little t, it's the only way to have that recessive trait. Brown as little b. So again, if I put them all together, they're kind of already close enough that I can just FOIL. I'm going to have little t, little b, little t, little b, little t, little b, and little t, little b. For some people, that's the hardest process. Part of the process is figuring out what the parents are, because you got to figure out what it is that you need to put here for the one parent and here for the other parent. Once you figure that out, it's usually not that difficult. So, it's just doing a Punnett square the way you would do them. So, when I say the way you would do them, you have to be careful. You're going to put the T's together. So, uppercase comes first, then lowercase. And you're going to put the B's together. So, again, even though we're dragging across and over, the order matters in the sense that you want to do upper and lowercase. Upper first, then lowercase. So if I'm looking at the next box in the sequence here, if I'm looking at this box here, then I've got a big T that I need to take, and let me switch there, a big T that I have to bring over, and a little t, and a little b, and a little b. And then I just continue this process. So here's big T, big B, little b. This is big T, little b, little b. And I continue my next row. Big, little, little. And you might notice, like sometimes you'll figure out there's a pattern. So in other words, this whole parent, their genotype is little and little, like little t, little b the whole time. So this actually means this whole row will be the same. I do allow you to do shortcuts up to like a whole row of 4. So in other words, you could just do this because you recognize that entire, well, I guess you'd say column is the same. And actually, that'll be the same for all the rest. Because this is all on the left side, the parent is recessive for both alleles, actually all the rest would be the same. But let's say I'm not going to do that. Let's say I want to write them out. Then again, if you wanted to write it out, like I'll go with the one I just erased, then, you know, you just have to look at each one, right? You would drag down the big T, drag over the little t, drag down the little b, drag over the little b, right? So you would just simply write in there, you've got big T, little b, right? But again, since I see it's the same there, I could just draw the arrow. This is an easier Punnett square though, because it's going to be 4, like each column is the same. But sometimes they're all mixed up and it's really difficult and it's complicated. So what I tell students to do is when you filled out your whole Punnett square, let's say each of these 16 squares, let's say that, I mean, sometimes there's like a 9:3 to 3:1 ratio. Sometimes it is a 4:4. I don't know how many 4s I said there, but sometimes it is like each 25% each, but it's not necessarily set up in these rows like this. So what I suggest to students to do is to kind of come up with a marking system. So what do I mean by that? If I'm analyzing my first box and I see big T, big B, little b, what I tell students to do is put a little mark in that Punnett square, like some kind of symbol, big, little, big, little, meaning I drew a green dot to represent heterozygous for both traits. So every time you see a box with that same genotype, you put that same symbol. Then, you know, you go to the next box that you look at. All right, I see big T, little b. Okay, so let's do it like a triangle. Right? And actually, if I look at the rest of my boxes, which I should have done for my other one, I would actually find out that I have, in this case, it's not actually, initially I was just looking at the 4. It's not really going to be a 4:4 ratio. It's actually 8 and 8, right? Because if I actually look at my whole Punnett square, then I realize that 8 of them, so my genotypic ratio is actually 8 big T, big B, little b, to 8 big T, little b, right? And what you got to be careful with is your phenotypic ratio, because you have to separate these out individually. So you might say to yourself, well, isn't it the same phenotypic ratio as well? But it's really not. So you look at the T's separately and the B's separately, or meaning your 2 different characteristics separately. They do happen to both have that tall characteristic, a big T, but they differ in the fact that the one has the dominant allele, it's black in color, but the other one, well, other ones I should say, since there's 8 of them, is recessive and has brown color. So with that in mind, we have to be really careful how we do our phenotypic ratio. So when you're writing out your phenotypic ratio, you're going to have 8 that are tall and brown. I'm sorry, not brown. Dominant was black. Tall and black. But of course these down here will be brown because they are lowercase, but meaning that it would be to 8 tall, and brown. So when you're writing out your ratios, you just got to be careful, because you have to look at each characteristic separately and figure out which trait, if it's the dominant or the recessive trait. So hopefully this video helps. Dihybrids can be tricky to understand initially, but once you get the understanding down, then they actually kind of can be fun and easy. Hope this helps.